Hello dear students, in last lecture we have discussed about the role of microorganisms in role in organic and inorganic matters. Now we will discuss about the again one of the topic, that is what is in the pure culture concept and what is the need to isolate the microorganisms in a pure form. So when we talk about culture is nothing but it is a growth of one type of organism. In case of bacteria, we call as the is a colony of for bacterial growth is we can call as a colony. So when it is a multiple type of organism can be found in that culture, we can form it is a mixed type of culture. And if we isolate that, we have to study that particular organism. So we need a pure single only one type of organism, and that culture is called as pure culture. So what is mean by pure culture? We will discuss. Microorganisms occur in nature everywhere, as we have discussed, in extremely large population. Normally we talk about 1 gram of soil contain millions numbers of microorganisms. So in this respect, to study characteristics of a particular species or a particular individual organism, it is necessary or it becomes necessary to isolate that particular organism into a pure form. What is the need? When we discuss about the microorganisms, we have to discuss about their beneficial and harmful activity. To know about their beneficial and harmful activity, we have to study that particular organism deeply. To study that particular organism, we need that organism in a pure form, or to characterize that particular organism on the basis of their morphological, biochemical, physiological characteristics or their metabolic activity. We have to study. We need that particular organism in a pure form. So it becomes necessary to isolate that particular organism in a pure form. Laboratory procedure have been developed to isolate them in a pure culture. So as it becomes a need to isolate that particular organism in pure form, different methods to isolate a particular organism in pure form has been developed. And now in the syllabus we will discuss about the different pure culture isolation method, likewise stick plate method, pour plate method, spade plate method. These are the different culture methods we can use to isolate organism in a pure form. Apart from that, the growth mass of the cells of the same species is called a pure culture. When we talk about the culture, it is the growth of organism on a solid or liquid media, we can say. So it is nothing but the growth of that particular organism. It is a culture. But if we have a single only one type of organism, that organism or that culture is called as pure culture. Pure culture of bacteria was first obtained by the Joseph Lister in 1878 using the serial dilution technique in liquid media. One of the scientists, or we can say one of the microbiologists, whose name is Joseph Lister, he isolated the pure culture of first time pure culture of organism in 1878 by using the serial dilution technique. Nowadays we can also use serial dilution technique for the isolation of pure culture, as there is a multiple number of organisms can be present in a 1 gram of sample. So to minimize the quantity of particular population of microorganisms, we have to dilute that particular sample using serial dilution technique. So this technique used by the Joseph Lister, and 1878 he has isolated pure bacteria in a pure form. Robert Koch isolated pure culture of pathogen on a potato agar using gelatin as a solidifying agent. So when we are talking about the microbiology, it is below it is discovery are 18th or 17th century. So primarily potato agar are used to isolate the organism, and to solidify that potato agar, gelatin was used to isolate the pure culture or to grow the particular organism on a potato agar. So Robert Koch used that particular agar to isolate the organism or to grow that organism in laboratory condition. But many bacteria can digest gelatin. Later on, what happened, as he prepared a media by using gelatin, he has solidified that media and allow organism to grow on that particular media. But some of the organism has ability to utilize or digest that gelatin which is present in that media, so that as gelatin is used up by the organism, that media becomes liquefied or there is no proper growth can be observed. So it was the drawback for that media. Later on, one of the colleague of Penny Hesse, or one of the assistant of Robert Koch, that is Penny Hesse, a wife of Walter Hussey, and he was the assistant of Robert Koch. He suggested the use of agar-agar for the solidifying agent, as it is used in the jam and jellies. So he suggested the use of agar as solidifying agent. Later on, agar-agar was used for the solidifying agent for the preparation of media, and they found that it is quite effective over gelatin. So agar-agar or agar is nothing but a polymer which is composed of D-galactose 3,6-anhydro L-galactose and D-glucuronic acid extracted from the red alga, that is Giladium corneum. So it is a little bit chemical structure of agar powder or agar-agar, and it is extracted from the red alga from the sea. And name of the red alga is Giladium corneum. And that agar is extracted from that alga was used as a solidifying agent over gelatin, and they found that the agar is more effective over gelatin. In what aspect? The agar was well suited as solidifying agent, as it solidifies after boiling at as solidifying agent after it boiled 42 degrees Celsius, and will not melt again until the temperature rises to about 80 to 90 degrees Celsius. So what happened when agar on the place of gelatin was used? They found that it start boiling about as temperature rise up to 42 degrees Celsius, and while we have prepared a media and that agar is when solidified, what happened? To again melt that particular agar which is present in the media, we have to rise the temperature of above 80 to 90 degrees Celsius. So these are the benefits of gelatin. So they found that it is quite effective again as a solidifying agent. One of the Koch assistant, Richard Petri, developed a Petri dish, a container for a solid culture media. So it is again another discovery of that time. They found that Richard Petri developed a Petri dish for the isolation or growing the particular organism on a Petri plate. So agar-agar as a solidifying agent and Petri dish was developed at that time, and they are quite useful to found. They found that to isolate the pure culture, that in the diagram you can see it is nothing but a Petri plate. The Petri plate size are fixed. It has a diameter about 9 cm and height about 1.2 cm. So it is a plate, it is a lower lid, you can see it is upper lid. We can pour our media on lower lid, and we have to allow that media for solidification, and after solidification we can strip your sample by this way, by strip plate method. So in that diagram we can see a pure well isolated polymer we can easily isolate. So it is all about the pure culture concept. So what is the need and how that pure culture can be isolated. So in this diagram you can see it is a serial dilution technique. So here in first tube we have taken a serial saline solution, and in that we have added a 0.1 gram, or you can say 1 gram, depending upon the sample from where we have taken, depending upon the population of microorganisms which are present in that sample. So we have to inoculate your raw sample into that saline solution. You have to properly mix that sample into that particular solution. After that, from that either from 0.1 ml, 1 ml or 2 drops of that sample can be transferred to another tube which contains saline solution. So what happens? The representative flora of that particular sample can be transferred to second tube. So dense population we can avoid here, so that the population density of microorganisms which are present in that soil can be minimized. After that, we have to add that particular sample, drop by what you can say 1 drop or 2 drop on a solid already prepared solid media at the center of that plate, and by the use of bent glass rod we can spread that culture over the surface of that media. So what happens? The proper distribution of microorganisms can be carried out on that surface, and each organism of that sample getting a proper nutrition, so that they start multiply and they form is well separated and isolated colony on that particular media. It is nothing but a spread plate method we can say, and it is also a serial dilution technique. Apart from that, we can also use strip plate method or core plate method also for the isolation of pure culture. So it is all about pure culture concept. Another concept is nothing but the aseptic surgery we can also use nowadays, and already we have discussed here Joseph Lister how isolated the organism, and he found that as use of phenol can be useful to avoid the infection of wound of the patient. So he used phenol for dressing the particular patient bandaging, and he used heat sterilization of the instrument. So what happened during the surgery, all the instruments are properly sterilized so that no contamination can be found, and we can avoid the entry of unwanted microorganisms into the wound so that other infection can be avoided. And apart from that, application of carbonic acid or we can say phenol to wound by means of dressing. So if we have added phenol on dressing or the bandage, it can also block the entry of microorganisms and can be helpful to prevent the other infection which can be caused by the other organisms. So it is quite effective technique given by the Sir Joseph Lister, and that technique is named as a aseptic surgery. That point we have already discussed. So it is all about the aseptic surgery and pure culture concept. Next point we will discuss in next.